Hi, good evening, everybody, and thank you so much for coming to tonight's lecture. And hello, everybody who's watching online. We really hope that there'll be people all over the world who are engaging in tonight's lecture and will be able to ask questions later on. So, all very exciting. In the past 20 years, phenomenal progress has been made in unraveling the complex chain of events that lead to the damage and loss of dopamine-producing nerve cells in Parkinson's. Those insights provide great opportunities, and exciting progress is being made to develop those into new treatments that can slow, stop, or reverse Parkinson's. But we at Parkinson's UK don't believe that fundamental science is sufficient to lead to the new treatments that people living with Parkinson's so desperately want. What people say to me all the time is, come on, Steve, we need treatments faster. We need treatments in years, not decades. So that has forced Parkinson's UK to really think about how we approach research. Yes, it's really important to fund university research and to look for those breakthroughs, but that's not sufficient to see the treatments that everybody so urgently requires. So that's why we've changed our approach towards drug development, and that's why we launched our Parkinson's virtual biotech at the start of this year, and that was the theme of last year's lecture. In the lecture program, you can read all about what that virtual biotech program is all about. Because through that, we really do mean business. By this Christmas, we'll have 2 new drug development initiatives underway, and that's representing around £2 million worth of funding that so many people in this room actually have made possible and can feel really proud of. But based on the success of this year, we're really ambitious about next year, and we're setting ourselves the target of significantly increasing our research spend. We want to double the amount of money that we're spending on the virtual biotech, and we want to initiate a further 5 projects next year, including a clinical trial. So that's all very exciting. But one of the challenges that we have in developing new treatments is actually the clinical trials themselves. And many people in this room will have seen with real disappointment the way that so often a promising clinical trial ends up not meeting its endpoints and being abandoned. That's a massive, massive disappointment to people affected by Parkinson's, but it's also a big disincentive for pharma companies investing millions of pounds in then maybe we should be investing our money somewhere else. But what if the problem isn't actually the drugs themselves that are being tested? Maybe it's not that the drugs don't work, but maybe we're testing them in the wrong way. Maybe it's actually the clinical trials that are not fit for purpose. So that's the reason why we launched the critical path for Parkinson's to try and make that process of undertaking clinical trials more effective. So we launched this international initiative that brings together researchers from all over the world, international drug companies, and regulators, so together we can change clinical trials so that they're smarter, faster, and more likely to succeed. It's that last piece in the jigsaw that's vital if the initiatives that come through from our virtual biotech program, but also the development programs of global pharma companies, they need that last piece in the jigsaw to make sure that clinical trials are more effective. So from our perspective, the science is ready, we have the expertise, and now new treatments are within our grasp. People with Parkinson's need a breakthrough, and we won't wait any longer. So that's the theme of tonight. How can we make clinical trials more effective so that we can bring forward the day that we are introducing those new treatments for Parkinson's. And to help us understand this, we've got four fantastic speakers here to talk about how we're doing that. First of all, well, not first of all, actually, Professor David Dexter, our Deputy Research Director, who's just recently joined us from Imperial College London, where he was Professor of Neuropharmacology and the Scientific Director of our Parkinson's UK Brain Bank. So fantastic that David has brought all that expertise into the charity. And then two jet-setting international partners who have flown in for this talk. First of all, Diane Stevenson from the Critical Path Institute, who leads the Critical Path for Parkinson's project. And as you'll hear, brings not just bags of energy and enthusiasm, but real expertise in how we can work with pharmaceutical companies in a consortium to achieve those objectives. And also delighted to welcome Jesse Cederbaum, who leads on early clinical development work in neurology at the pharmaceutical company Biogen. So it's going to be fantastic to have an industry perspective on why this work is really important. It's wonderful that Jesse is joining us to share his insights on the impact from an industry perspective. And we know that he's so jet-setting that actually he'll be rushing off halfway through the meeting to, I'm sure that's for an urgent reason. There must be some good Parkinson's drug development discussion that's taking place tomorrow, or I hope so anyway. Otherwise, you haven't got a good enough excuse. But first, and I think to really set the scene, I want to introduce Leslie Gosden. Leslie recently took part in a clinical trial. She'll share her experiences of that and set out really clearly why we so urgently need to improve the clinical trials process. So, Leslie, thank you.
Thank you. First of all, good evening to everybody, and thank you for inviting me to speak to Parkinson's UK. 14 years ago, I was 43, had a young family, a responsible full-time job, financial security, and a good work-life balance. Then I was diagnosed with Parkinson's. Today, whilst my two sons have blossomed into young adults and are thriving, I've taken ill health retirement, finances are tight, and my health is variable. Disease progression for me has been thankfully slow when compared to fellow sufferers, but the condition is relentless and there is no respite. I wake at around 4 to 5 a.m. each morning with my whole body stiff, aching and inflexible. My brain is unable to coordinate my body, and rolling over in bed is just impossible. I feel lethargic, bereft of any energy and utterly exhausted. Getting out of bed is a nightmare, and achieving my goal of reaching the bathroom is challenging. I know sleep will now elude me as I'm wide awake, so I shuffle to the spare room where I will endure several hours of discomfort before taking medication at 7:30 a.m. Any earlier, and I'll face increasing symptoms through the evening. There is a finite limit to the amount of drugs I can take, and increasing my intake at this stage is avoidable. Each day starts like this. Every day of the week, every week of the month, and every month of the year, and in the certain knowledge that, as this is a degenerative disease, each day will be a little worse. Until a cure is found, this is a life sentence. The discomfort, pain, and sheer frustration of coping with Parkinson's, whilst coming to terms with its wanton destruction of the life I once knew, fueled my determination to take an active role in research. The purpose of the GDNF clinical trial was to determine whether infusing a protein which was designed to revive dying cells into the affected part of the brain could slow, stop, or even reverse symptoms. Participation required risky brain surgery, which had never been performed before, to insert a delivery system that had never been tried before, that would infuse experimental fluid into the brain, and to commit to 2 years of monthly assessments. Every other assessment had to be in the off state, which meant stopping all medication 24 hours in advance. I fear the future and what it holds, and dread the possibility that I might one day become a burden on those I love. Not how they see it, I know, but I have their freedom and my dignity to consider. I also worry about the possibility that my sons could develop Parkinson's and would do anything in my power to secure a better future for them than I currently face. GDNF offered hope, just a glimmer, but hope all the same. I applied, and following intense clinical and cognitive tests, was accepted. The delivery device was put into place during 6 hours of surgery, assisted by robot, using a 3D plan of my brain. This had been constructed from a 3-hour session in the MRI scanner and was to ensure avoidance of blood vessels and other areas of the brain that might object to having the small tubes inserted. The variety of symptoms and stage of disease varied enormously amongst the participants. For example, I travelled in a full taxi from Bristol to Cardiff. I was unmedicated, and almost immediately my legs and feet went into spasm. The other patient in the car quickly volunteered to sit in the front seat and slided as far forwards as possible to give me more room. His height meant that he had to tuck his knees right up under his chin for the duration of the journey. So whilst confinement in a tight space results in cramping for me, he had no ill effects. Cardiff was the location of the PET brain scanner, the part of the trial which many participants found difficult. After injecting radioactive dye, I was strapped to a narrow board with my head secured in a tight frame, then pushed headfirst into a tube that was just about large enough for the average adult. I was then expected to lie completely still for over 1.5 hours, or to be precise, 95 minutes. That's 5,700 seconds. The lack of my usual drugs meant that cramping had already taken hold in my toes before I even entered the scanner. By the time I was fully enclosed, I was terrified, and total panic set in. The spasms continued to spread from my toes, through my feet, calves, thighs, and into my lower spine. The pain was unbearable, absolute torture, and I begged to be removed. I'm still haunted by the experience and frequently wake in the night in a cold sweat, shouting to be let out. Thankfully, subsequent scans were performed with the aid of sleeping tablets and a muscle relaxant. At this point, I'd like to take the opportunity to thank Alan Wone and his incredible team. I have nothing but gratitude, admiration, and total respect for his clinical and administrative staff, whose care and support during the trial has been exemplary. They've been supportive, professional, and utterly dedicated. This was a double-blind trial. Half of the participants received placebo and half the real thing for the first 9 months. Neither the participants nor the clinical team knew who was receiving which fluid. For the remaining 9 months, all participants received GDNF. As some of you may already know, the initial top-line results revealed that the trial missed its primary endpoint, or in plain English, when measured off their regular Parkinson's medication after 9 months, those who received GDNF were not significantly better than those who received placebo. The early stage of the trial, along with the placebo effect, has to be taken into account. Regular NHS care consists of 9 monthly consultations, during which time disease progression is assessed, medication reviewed, options considered, and notes taken, all squeezed into 10 minutes at most, and usually less due to the late running of clinics. Contrast this to the trial experience: monthly checkups and medication review with a 24-hour dedicated helpline could induce a feeling of well-being amongst participants and potentially ease symptoms. During the trial, I experienced a gradual improvement in symptoms. Today, the excruciating frequent cramps and muscle spasms, which left my feet and toes curled into unimaginable shapes, have reduced considerably. I can walk more quickly and much further. I have the confidence to drive longer distances, and overall I feel more alive and energetic than I have done for some time. Family and friends have commented on my new lease of life and how much better I'm coping. I can feel the benefits of this trial in every breath and every movement. So why are we not planning the next stage of the trial instead of anxiously awaiting news on the future? For me, the answer is simple. The tests performed were artificial and did not reflect how the symptoms affect everyday tasks, nor did they assess those symptoms which had the ability to improve quality of life. For example, I was questioned about dystonia, that's muscle spasms, and was timed walking across a small room. But there were no physical tests to measure the reduction in frequency or strength of the spasms, my ability to walk much greater distances, or my increased stamina, all of which have a huge impact on everyday living. I met many of my fellow participants, and whilst I cannot speak for them all, the overall feeling was positive and hopeful, and the fact that no one gave up or dropped out is testament to the belief that this was promising. Each individual patient has their own specific set of symptoms. There is no common denominator. Despite this, we are still using one-size-fits-all tests on a disease that treats protocols and standards with utter contempt. It manifests itself in a myriad of guises and can morph into a different presentation depending on outside influences like stress, constipation, fatigue, and so many more. Understanding the devious nature of Parkinson's is crucial in the fight against it. We cannot expect it to cooperate with the regimented, inflexible parameters by which it is assessed. The initial diagnosis of participants also needs careful scrutiny, as other presentations mimic Parkinson's, and it has been shown on autopsy that many patients are misdiagnosed. Innovation in research demands an in-depth understanding of the function of the brain, imagination and dedication to turn these ideas into projects, and the awe-inspiring skills of the clinicians and surgeons to make them a reality. I owe a huge debt to the network of scientists who are beavering away to find a cure for Parkinson's. I do not understand the disease scientifically. For that I rely on them. I do, however, have an extensive knowledge of living with Parkinson's. Clinical trials are initially carried out using mice. The progression to human subjects opens up the opportunity to harness their unique ability to assess and describe how they feel. Despite this, throughout the trial, no one has asked for a summary of each participant's experience. We've been asked a series of questions which require tick-box answers, but there is no scope for variation or elaboration. An absolutely crucial resource has been overlooked. In order to find the elusive cure, we need to get better at clinical trials by learning from our mistakes and creating better tools and strategies that maximize our chances of success. Otherwise, millions of pounds will be spent pursuing projects which could be the next big breakthrough, but will remain unproven. That's why I'm so delighted that Parkinson's UK have formed the Critical Path for Parkinson's to bring the right people together to make this happen. When I first joined the GDNF trial, it was made crystal clear to me the difference between hopes and expectations. I could hope for a good outcome, but I should not expect one. Taking part offered no guarantees. I still completely understand and accept this proviso, but at the same time, whilst I can only hope for success, I should be able to expect that should there be just the merest glimmer of hope, then the criteria by which results are judged are robust enough to reflect this. The smallest chance that a treatment could work should be seized upon. Clinical trials are essential in the process of discovering new and repurposed treatments and are reliant on volunteer patients. Personally, I would have absolutely no hesitation in taking part in further trials and will keep going till we succeed. But the risks of trials have to be weighed against the chances of success, and if current trials continue to fall short of their targets, contradicting overall perception, recruitment will be challenging. This trial has been emotionally, mentally, physically draining. It's certainly not for the faint-hearted. Putting my life on the line was no easy decision. It took the brave and loving support of my family, belief in the scientists, faith in their clinicians, and a desire to beat this cruel, debilitating disease. This was a huge commitment, and having endured the rigors of the trial, rediscovered my zest for life, and been reminded of how my body used to feel, I am anxious to continue with the next phase. The realization that medical science has yet to prove its benefits conclusively has to be the most frustrating and distressing experience of my life. The extended wait for results, which were initially expected in the spring, has put my family and I under tremendous strain as we anxiously wait to hear the future of this trial. The next big breakthrough always seems to be 5, 10, or even 20 years in the future. I've endured Parkinson's vindictive desire to destroy my quality of life for over 14 years, and quite frankly, I've had enough. I and thousands like me do not have the luxury of time on our side. We want hope, and we want it now. Thank you. Thank you very much. I'd like to introduce the next speaker, who's Professor David Dexter, a Deputy Director of Research.
Good evening, everybody, and thank you for giving me the opportunity to actually talk to you this evening about some of the interesting developments taking place in the Research Directorate of Parkinson's UK. There are currently about 127,000 people affected by Parkinson's in the UK at any one time. And it's 200 years since James Parkinson first described the disorder. And the treatment strategies for treating Parkinson's really haven't changed very much ever since the development of L-dopa, and that's 50 years ago. So current drug treatments only treat the clinical symptoms of the disease, but they don't slow the disease down. So there is no cure at the moment, and so we desperately need better treatments and better cures. So traditionally, late-stage development of new drugs takes place with pharmaceutical companies, but due to high